Can you hear me? Good. Maybe, okay. So thanks, Raj. Maybe the microphones. There's a lot of people. Okay. Okay, I think that's better. Okay, so I have two different sets of things to talk about, but we'll also have questions along the way. So the overall theme will be the role of RNA metabolism and RNA binding proteins, RNA processing pathways, and how they affect neurodegenerative diseases. So if you have questions, if you want to talk about something else or you want to clarify something, just please interrupt me. I'll have some questions along the way. So my laboratory studies neurodegenerative diseases, and these are increasing in prevalence as the population is aging. And these are very different types of diseases. So like Alzheimer's disease affects memory. This is like a series of self-portraits from an artist as he sort of declines with his ability to perceive himself through Alzheimer's disease. So that disease affects cognitive ability, but Parkinson's disease is a movement disorder. Cognitive ability is relatively preserved. So the idea is that different neurodegenerative diseases have different clinical features, but despite those differences in clinical features, all of the neurodegenerative diseases, the major ones, are associated with protein misfolding. So proteins in the brain of patients with these diseases become misfolded, and then they start aggregating and accumulating in the brain. Sometimes they can accumulate outside of neurons, like Alzheimer's disease, amyloid plaques, and sometimes they can accumulate within neurons, like neurofibrillary tangles and frontotemporal dementia. They can be in the cytoplasm, like in Parkinson's disease, and they can even be in the nucleus. These are polyglutamine aggregates in Huntington's disease. So the idea is that different proteins misfold in different parts of the brain. It's also, I think, a very interesting basic cell biology question because these proteins and the genes that are encoding them are expressed throughout the brain. Some are even ubiquitously expressed. Yet the diseases have very selective vulnerabilities. Some types of neurons are lost and others are not. So I think this means that these proteins, and when they aggregate, they interfere with certain cellular pathways. I think this means that these diseases, even though complicated, are rooted in basic cell biology. So I'm interested in understanding the cell biology of these protein aggregates and what forms them and maybe the consequences of that. Today, as an example, I'm just going to talk about one neurodegenerative disease, but it's just an example, and hopefully you can see the types of approaches we're taking and questions we're asking, so you can see how that can be applied to something you might be interested in. This is a disease called amyotrophic lateral sclerosis, or ALS. In the United States, it's called Lou Gehrig's disease. He was a famous New York Yankees first baseman. This is a disease of motor neurons. So neurons in the brain and the spinal cord, they innervate muscle. And when they die, it causes muscle weakness. And then that progresses to paralysis. And then very rapidly, it's fatal due to respiratory failure. You can see that the disease can have variability. So it affects individuals when they're younger, or individuals can live, it's a very rapidly progressing disease, or individuals can live for many years with the disease. So it's very heterogeneous. Over the years, genes have been identified as causing ALS. So about 10% of all ALS cases are caused by mutations in specific genes that inherited from one generation to the next. And identifying the genes that are causing those forms of the disease provide insights into mechanisms because they can give us pathways to think about. Like the other neurodegenerative diseases, ALS is a protein misfolding disease. So this is a section through a spinal cord of an individual with ALS, and you can see that there's proteins that are aggregating. About 2% of all ALS cases are caused by a mutation in a gene called SOD1. And if you have SOD1 mutations, the protein that aggregates is mutant SOD1. But for the 98% of cases that do not have the SOD1 mutations, they also have aggregates in the spinal cord and the motor neurons, but it's not SOD1, it's another protein. And that protein was discovered in 2006 by my colleagues in the University of Pennsylvania, Drs. Virginia Lee, John Trochanowski. They found that this was an RNA binding protein called TDP43. It's normally nuclear. It binds to RNA, it regulates splicing, but then somehow in disease, it gets depleted from the nucleus and it accumulates in the cytoplasm in these round, Lewy body-like, or these ropey, scheme-like aggregates. And this is the major pathology seen in virtually all ALS cases. Very rapidly after they identified the TDP-43 pathology, geneticists came along and they identified mutations in the gene that encoded TDP-43 as a cause of some rare forms of ALS. So now it's this combination of genetics and pathology that's focused everyone, and probably why I'm here at this meeting, because it's an RNA binding protein, focused everyone on trying to figure out how TDP-43 might be contributing to ALS. So maybe one question for you to think about is how does TDP-43 and or mutations in the protein contribute to disease? Here are some clues. So this is a map of the protein. This is RNA recognition motifs. Then this is the C-terminal domain. And almost all of the mutations are here. And then on the bottom are two sections of human spinal cord. I'm going to show this picture a few times today for different reasons. So here, this is a large motor neuron. This is the nucleus. You might be able to see the nucleolus. I stain this. This is from a human spinal cord. I stain this with an antibody against TDP-43. And this is the normal situation. You can see TDP-43 is diffuse. And then this is the disease situation where TDP-43 is here. So what do you think are some ways in which TDP-43 might, or TDP-43 pathology of the mutations might contribute to disease? Mislocalization, so from where to where? Okay, good. So it seems to be, it is in the nucleus. And now here, it's now in the cytoplasm. And if you look here, the nucleus is kind of empty. So it's mislocalized. So it's not where it's supposed to be. So it can't carry out its normal function. But you can also, and you also said aggregation. So that's also true. Now we have these large aggregates out here. This is not the nucleus, by the way. This is a large clump of TDP-43. There are also these smaller brown dots. Maybe these are some sort of liquid accumulations. So maybe TDP-43 is causing disease by aggregating and interfering with RNAs and RNA binding proteins out here. Or maybe it's caused by it not being where it's supposed to be. Or maybe there's some combination of both. They're not mutually exclusive. So I think sort of a theme today will be considering the potential roles of some sort of cytoplasmic gain of toxic function due to aggregation and some sort of loss of function from the nucleus. So in my laboratory, when I started my laboratory, I wanted to figure out how TDP-43 aggregation might be contributing to disease and what's causing it and what is the consequence. And as a postdoctoral fellow, I use the yeast as a model system. You can, protein aggregation is conserved and all of these major cellular pathways are conserved. I wanted to see if I could use this system to study protein aggregation. It's very rapid. So whereas the disease in humans takes decades, I'll show you some experiments where I could put human aggregation proteins from disease into yeast and it'll start aggregating in just a couple hours. And then it will elicit phenotypes. And then we can use those phenotypes to screen for modifiers. And then we can go back and forth and validate our findings from yeast into these other model systems. And then recently we've expanded our analysis to human patient samples and mouse models. I'll show you some of those examples. The power of yeast is that we can perform unbiased genetic screens. You can now do this with CRISPR, but it's real in mammalian cells, but it's really straightforward in yeast. There's 6,000 genes, and about 4,850 of them are non-essential. So, and we have deletions in our freezer of every single non-essential gene. We have ways of interrogating the essential genes with conditional alleles. We have a library of all yeast open reading frames that we can overexpress one by one, and that's what we did. We have our yeast cells that are about to express TDP-43 in yeast, and it's going to be toxic. It's going to start killing yeast. And I want to see, can I find genes that can make it better or worse? This is, the logic is like, I have no idea why TDP-43 is toxic in a human cell or in a yeast cell when it aggregates. But let's just say TDP-43 inhibits some essential enzyme in the cell. You can imagine in a screen, if you overexpressed more copies of that enzyme, you could suppress the toxicity. You can imagine if you put more copies of like a negative regulator of that enzyme, you might make it worse. So if you do the right controls, positive and negative controls, suppressors and enhancers of toxicity are both informative. So we have in our laboratory these robots where we can take our yeast cells and spot them onto large agar plates and then pin them over. And you can do a whole genome-wide yeast screen and triplicate in like 3 weeks. So this is what the yeast cells look like. This is a yeast cell, its daughter cells budding off. And when I expressed TDP-43 in yeast, I fused it to GFP. It formed these liquid-like inclusions. This was back before we knew what liquid-liquid phase separation was, so it just seemed like they're just green foci in cells. And then when we did the spotting assay where we took yeast cells and serially diluted them 5-fold, spotted them to agar plates, I did this in a way where I could only express TDP-43 when they were grown in the presence of galactose. This is a galactose-inducible promoter. When I was growing them in the presence of glucose, it repressed expression, just to show you that I spotted the same numbers of cells. If they express GFP, they can grow, they form colonies, no problem. But when they express TDP-43, it was highly toxic. So this recapitulated 2 of the salient features of TDP-43 relevant for disease, the aggregation in the cytoplasm, and then this toxicity. And this is an example of one of the plates from our yeast screen. We have 96 spots per plate. Each spot expresses our yeast cells expressing TDP-43, and then overexpressing 1 gene from the yeast genome, and we do all 6,000 genes. We do this in triplicate. Most had no effect, but you can see like this guy allowed the cells to grow even in the face of TDP-43. So that would be like a suppressor of toxicity. These guys made it worse. So these would be enhancers of toxicity. A little worry I had when I started this in my own laboratory was that I'm putting a foreign aggregation-prone protein in yeast. And it's going to have all these unspecific effects. And when I was a postdoctoral fellow at the Whitehead Institute with Dr. Susan Lindquist, I did the same screen, but I did this for this other protein called alpha-synuclein. This is a protein that aggregates in Parkinson's disease. And I was just thinking, in my own lab, I'm just going to get all the same stuff I got as a postdoc, and it wouldn't be informative. But what was cool was even though alpha-synuclein and TDP-43 look similar when you express them in yeast, doing the same screen, same library, same robot, me doing both screens myself, same everything. I get virtually non-overlapping hits. So this is, I think this is exciting because this means that we're not just getting unspecific things due to protein aggregation. I think it means that we're hitting at the underlying biology of the disease protein. So that specificity really gets me excited. So we have about 40 genes that when overexpressed in yeast modify TDP-43 toxicity. And whenever you do a screen, like a yeast screen or a Drosophila screen or mass spec, RNA-seq, single, whatever, you always get a list of hits. Like any of you in your projects, you get lists of hits. And it's always a challenge to figure out like which hit on your list will be informative. So I just want to share with you one hit we worked on because it took us in a lot of different directions. So you just can see how we approach these things. It's a gene called PBP1. We found that when we overexpressed this, it enhanced TDP-43 toxicity. It made cells expressing TDP-43 grow slower. This is wild type cells expressing TDP-43. If we also overexpress PBP1, they grow even slower. It was specific. It didn't make cells expressing mutant Huntingtin or alpha-synuclein grow slower. And then on the other hand, when I deleted PBP1, this did the reverse. Wild type cells, TDP-43 is toxic. Delete PBP1, it suppresses the toxicity. So this means it's a dose-sensitive modifier. Upregulation makes TDP-43 more toxic. Deleting it makes it less toxic. Of the genes on the list, other than the fact that it had a strong effect and went in both directions, I focused on PBP1 because it had a human homolog that had already been connected to another human neurodegenerative disease called spinocerebellar ataxia 2. The gene's called ataxin 2. It's one of these polyglutamine disease genes like Huntingtin. And then we collaborated with my colleague at the University of Pennsylvania, Dr. Nancy Bonini. Luckily for us, she had all these flies that could upregulate ataxin 2 or lower its levels. And we had been making with her flies to do the same thing with TDP-43. So we just said, hey, let's cross the flies together. In this experiment, we take flies and we direct the transgene to the fly eye. And on the top is a picture of the fly eye, and the bottom is a section through the fly eye. Overexpressing TDP-43 by itself caused a mild degenerative phenotype, no effect over expressing ataxin-2. But when I combined the two together, it causes this massively degenerating eye phenotype. And then when I lower ataxin-2, in flies, ataxin-2 is essential, so you can't get rid of it. But if you lower it by a half, this is suppressing the eye phenotype. Now, if we just direct the transgenes and express it throughout the whole nervous system, the flies live shorter when they express TDP-43. They don't live shorter if you overexpress ataxin-2 by itself, but together with TDP-43, they live even shorter, and then we can extend survival by lowering ataxin-2 and TDP-43 expressing flies. So that got us thinking about the ataxin-2 gene. I said it's a polyglutamine disease gene. What that means is that in the protein, there's a polyglutamine domain encoded by CAG, that encodes glutamine, abbreviated Q or poly-Q. And there's always 22 of these, sometimes 23. If it gets expanded to greater than 34, it causes this disease, spinocerebellar ataxia 2. But because I found this in a yeast screen, I was wondering, could this somehow be connected to ALS? I found it in a yeast screen with TDP-43. And so this is normal, 22 or 23. Greater than 34 causes this other disease. I was just thinking, what about something in between? Longer than normal, but not past that range for SCA2. That's my hypothesis. So to test that hypothesis, I collected about 1,000 DNA samples from patients with ALS, about 1,000 DNA samples from healthy individuals. No one in my laboratory wanted to do this, so I did this experiment by myself. I just ordered PCR primers flanking this region, and then just amplified this. This is a gel picture from my lab notebook. I would just cut these bands out of the gel and TA clone them and then sequence them, and I could just count the CAGs. Eventually, I was able to do a way where when you order your oligos from IDT or something, you could ask them to put a fluorescent dye in one of the primers, and then you could separate the amplicons by capillary electrophoresis, and you could see very precisely the size. So everyone has 2 copies of each gene, one from dad, one from mom. This individual up here, there's 1 peak. Both copies have 22 CAGs. This one, there's 2 peaks. One is 22, one is 23. But for the ALS cases, I started to see expansions at 27, 30, 32, 33. This summarizes all of the numbers. Out of 980 controls, I only found 14 healthy individuals with repeats greater than or equal to 27. But almost 5% of the ALS cases have these ataxin-2 expansions, these intermediate length ataxin-2 expansions. 5% doesn't seem like a lot, but it's actually now one of the most common genetic risk factors for ALS. So I think it's cool that it came out of a yeast screen. So it underscores the importance of simple model systems for studying, for learning things about human disease. And it's also, now we're sort of obsessed with ataxin-2. How does that contribute to ALS? So that's the question. I don't know this answer, but hopefully we can discuss it, and maybe, because it's the next step in my laboratory, is how do ataxin-2 poly-Q expansions cause ALS? Because we have to figure this out so we can figure out what the mutations are doing and stuff like that. So I don't think I have clues, but 22 is the normal length. 40 or greater than 34 causes spinocerebellar ataxia 2, and then in between seems to cause ALS. One thing that was a little bit counterintuitive, just to tell you, is it's not like people that have SCA2 have both ALS and SAA2. It's a distinct disease, although there could be some overlap. So any ideas, the mechanism by which these poly-Q expansions might cause disease? Okay, say a little bit more. Okay, so yeah, so we have different ataxin-2 proteins with different length Qs. Good, and then what? Yeah. So maybe if the more Qs, the more aggregation, or it might burden quality control. Yeah. Okay, that's possible. But then how? Why? The weird thing is, why would it cause motor neurons to die if it's 30, but Purkinje neurons in the cerebellum to die if it's 40 or something like that. I don't know that answer. I think it could, because one interesting thing is the repeat can be interrupted with CAA, which also encodes glutamine, and there's different phenotypes if there's CAA versus CAG. So that suggests that it might have some pathogenic effects at the RNA level, like you were saying. So that's absolutely possible. Okay, one observation we had, we had cell lines from patients that had the some fibroblast lines or some blood cell lines from patients that had these different expansions. And I just did a stability assay where I just shut off translation with cyclohexamide. And then it seemed that the proteins that had the intermediate length polyglutamine expansions, the ataxin 2 was more stable. So there was more of it around than the ones that were the normal length. So I thought more of it around. And then I was also remembering from the yeast experiments, if I deleted ataxin 2, they were sort of resistant to TDP-43. So I thought maybe if I just lower ataxin 2 levels, that could have a therapeutic benefit. Maybe there's just too much. So you need to do, like, we wanted to then think, to test that, we wanted to do an experiment in mouse. So Lindsay Becker was a graduate student in my laboratory at Stanford, and she did this experiment. She's now a postdoctoral fellow with Cliff Brangwyn at Princeton University. And what Lindsay did was she took mice that were engineered to express TDP-43 throughout the nervous system. These aggregates of the nervous system, and these mice die very early. And then she knocked out a ataxin 2, and she crossed those mice, and she looked at mice that had 2, 1, or 0 copies of ataxin 2. And she found that the normal mice expressing TDP-43 all died very rapidly by day 30. But if she had half as much ataxin 2, the mice lived significantly longer. But the crazy thing was when she completely got rid of ataxin 2, the mice lived a lot longer, like over 300 days. And this was also able to get rid of the aggregates of TDP-43 in the spinal cord. So it seemed maybe that ataxin 2 is required for facilitating TDP-43 aggregation. She then did an experiment where we collaborated with a pharmaceutical company to develop a kind of a DNA-like drug called antisense oligonucleotide to lower ataxin 2's level. And we delivered this. This is a DNA-like drug. It binds to an RNA you designed it against. And then all cells have an enzyme called RNase H, which recognizes RNA-DNA hybrids and then catalyzes the destruction. So we delivered this into the pups at day 1, either one targeting ataxin 2 or like a negative control, like a scrambled sequence. This was sufficient to lower ataxin 2 RNA and protein. And a single administration of the ataxin 2 targeting one had also a dramatic extension in survival. Some mice lived over 150 days, and the other ones just all died by day 30. This is a video of mice. You're not used to looking at this, but this just shows you that the rescue can be very strong. These are 3 mice at day 20. 2 of them we injected with a scrambled negative control sequence. These mice are paralyzed, but the other one we injected with an ASO targeting ataxin 2. This mouse goes on and lives much longer. So we're excited that you could target this modifier gene and have a benefit with ataxin on TDP-43 mice. I'm excited about ataxin 2 because I was kind of mentioning this at the beginning. The TDP-43 pathology really is the universal pathology in almost every ALS case, except those that have SOD1 mutations or this other RNA binding protein called FUS. And then also about 50% of TDP-43, 50% of cases of a related disease called frontotemporal dementia have TDP-43 pathology. So we're trying to figure out if targeting ataxin 2 can be beneficial there. We're really excited about antisense oligonucleotides. These have really changed the therapeutic situation for ALS. One targeting a mutant form of SOD1 was approved by the FDA and is now available around the world. This is the amazing thing about this is that before this, it wasn't really known if ALS was a treatable disease, but now individuals who have SOD1 mutations are getting treated with this. There's So one targeting this mutant C9 ORF22, I'm going to talk a little bit about this, that had some trouble. Biogen, together with the other company, we didn't work with them on that. They tested our hypothesis in human last year, and they found, unfortunately, that in a 6-month trial, they didn't see a benefit for lowering ataxin-2. So we're still trying to figure out like why this ataxin-2 ASO trial didn't work. It's complicated when you have a person who already has disease and a lot of neuron loss, and you're delivering something to the nervous system, and you don't necessarily get a good knockdown. So we're trying to sort all of that out. Okay, I want to move on to another, perhaps the most common genetic contributor to ALS, because it sort of led to like a mystery about how it's working, and sort of thinking about RNA metabolism as well. So there was, do you know what genome-wide association studies are? A little bit, okay. It's basically a population genetics technique where you look at variations throughout the genome and in 2 different groups of people. You can do it for height or diabetes, whatever. In this case, you can take a large number, thousands and thousands of individuals with ALS, and thousands of individuals without, hope that they're geographically matched, and then look what variations are associated with ALS. And there was a association peak on chromosome 9, 9p21. And they identified that region. So it's sort of like saying, I know that the house is on this street, but I don't know what the house number is. You sort of know where the street or the neighborhood is. And because this was the largest, it seemed like the most common cause of ALS, but the gene wasn't known. It was an international search for this gene. So if I told you that the gene was somewhere on chromosome 9, how would you find the gene? What are some things you would do? Sorry. With a cell, okay, so you would truncate chromosome 9. How do you do that? But we don't know what we don't have a phenotype yet. We just have people with ALS. We don't know like a cellular phenotype yet. Any other ideas? You know, kind of it's on chromosome 9. Yeah, you could look up chromosome 9, see if any genes make sense. Like, is this a motor neuron transcription factor or something like that? They did that. That didn't work. Then they sequenced every gene, the coding region of every gene on chromosome 9. They didn't find, just because this was back before like genome sequencing was super cheap. They just sequenced the coding regions. That was like more bang for your buck. They didn't find anything. Then like one of the technicians in a laboratory in Florida was just looking at one of the sequences, and she realized that there was this repeat G, 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 C, C, G, G, C. And then she thought maybe that repeat, this was in a non-coding part of the gene, maybe that would get expanded. So what she did was she did the same thing I did for my paper, where I just made those primers flanking that region and just looked for the number. And she was lucky because in addition to the GWAS signal, she had a family that also showed segregation at this at chromosome 9. So she did that PCR thing that I did, and just focus on this part of the family. 9, father is 9, mother is 10. So open square is unaffected. Closed circle is the mother who has had ALS, and then 16, 17, 18 are three children. 17 and 18 are affected, and then 16 is unaffected. So if you look at the father, everyone has 2 copies, right? The father has the repeat length is 5 for the father and 8 for the other allele. So 5 and 8, they have 2 copies. And then the mother is homozygous. Both of her copies are 11, okay? So then they look at number 16, got 8 from dad, got 11 from mom. That checks out, right? This is just like Mendel, like high school, like segregation. Okay. But then the confusing part came when they looked at number 17 and 18. They got 5 from dad, but they didn't get anything from mom. So this kind of violates like Mendelian inheritance. So there's had to be some problem there, right? So she was very clever and she thought maybe mom wasn't homozygous for 11. Maybe one was 11, but the other one was so big, it was like way off the board so that like they couldn't get it. So she came up with a new way, a repeat prime PCR way and southern blotting. And she found indeed enormous expansions, like hundreds or thousands of repeats in this gene. And now this led to these 2 famous papers that reported the most common cause for ALS. The gene is called C9orf72. So if you just guess the gene, you never would have guessed this one because it just means the 72nd open reading frame on chromosome 9. We still don't really even know what it does. And it's a hexanucleotide, so 6, GGGGCC, and it's repeated many, many times. And this is the most common cause for ALS. So now the challenge is how do we figure out how these expansions cause ALS? Some clues. So here are some clues. If you look in cell lines from patients with these expansions, there are these RNA foci that build up in the nucleus that have these. And the other thing is this repeat is in the intron of the non-coding part of the gene. And it's a massive expansion and it's GC rich. So what are some potential mechanisms in which having this massive expansion could cause disease? What's aggregating? Okay, like at the RNA level, right? So RNAs could aggregate, and then what do those RNA aggregates do? Yeah, it absolutely could do that. It seems that these RNA foci here are in the nucleus, so maybe they can interrupt things in the nucleus, interfere with splicing factors, disrupt the localization of things that bind to that G4C2 repeat. Good. Yep. Yeah, it could disturb RNA binding proteins. Good. It's also possible that like having this huge expansion in the regulatory region of your gene might just disrupt the expression and it might decrease expression of that gene. So you might have like a haploinsufficiency. And indeed, if you look at patients, you can see they have less expression. So maybe the disease is loss of function. And the other thing that's complicated is this repeat also can get transcribed in the antisense direction, GGCCCC, and that can accumulate. So maybe those RNAs are doing something. And then it really became wild in that there's a third hypothesis. It turns out that these repeats can get translated in an AUG independent manner in all 6 reading frames, the sense and antisense. There's no AUG to start translation, so all reading frames get made to make like glycine alanine, glycine proline, glycine arginine, and then the other ones like another copy of glycine proline, proline alanine, and proline arginine. And if you make antibodies against these predicted dipeptide repeat proteins, they're aggregation prone, and you can see them accumulating in the brain. So maybe these proteins are causing disease by some toxic thing. So it gets pretty complicated, right? We still don't know how those are causing disease. And all 3 of these mechanisms are not mutually exclusive. And if you think about it, we have to figure this out for like thinking about therapies. We, just briefly before the break, I'll just show you, we made a couple insights, we went back to yeast. We found that you can detect the sense and the antisense foci when expressed in yeast, and we've sort of been using this yeast system to study for modulators of that unconventional form of translation called RAN translation. But then in meetings and things, people are always arguing whether it's the RNA or the protein, dipeptide repeat proteins that are toxic. So we thought we could use yeast to test that. So we made constructs to express just those dipeptide repeat proteins. We codon optimized them so there's no RNA repeat, and there's an ATG, so they just express things like glycine alanine, glycine arginine, the proline arginine. And we did this spotting assays you're used to seeing now. And we found that the arginine-rich ones were very toxic, like proline arginine, exceptionally toxic. At the same time we were doing this, other groups found the same thing, that the arginine-rich ones are toxic in human cells, in Drosophila, and then in human motor neurons in Drosophila. Like you can see, this isn't my experiment, but this just wipes out the fly eye if you express proline arginine or glycine arginine. So there's lots of bad things going on here. We want to find out like the key things. So this is why you do genetic screens. We went back to yeast. Can we find like modifiers of this? And this was performed by Anna Jovicek, a postdoctoral fellow in the lab. She's now a senior scientist in Genentech. Normally our screens are difficult. They all like, we have to come up with hypotheses and like mine data. With this one, it was very easy because they all were on the same pathway. They all converged on the nuclear pore. We got all the carrier proteins that transport proteins into the nucleus, components of the nuclear pore, the enzymes that power the RAN gradient. So we proposed that C9orf72 mutations impair nucleocytoplasmic transport. The same day we published Anna's paper, 2 other labs published the same results in another journal, which is cool if we all got the same results, but then we all disagreed about the mechanism. We said it's the proteins, these guys said it's the RNA, these guys said it could be both. Then these guys said it was the proteins, not the RNA, and we keep going back and forth. But I think the main picture is that now it seems that these mutations are somehow converging on nucleocytoplasmic transport. We've extended our studies to like mammalian neurons, and we found unexpectedly that the tumor suppressor gene P53 is required in neurons for toxicity from these dipeptide repeat proteins. The knockouts are completely resistant. Maybe I'll take a break, or yeah, I'll take a break, and then we'll go on to the next section. Because now we've been talking a lot about gain of function stuff. The next section is all going to be what happens when we lose TDP43 from the nucleus.
So do you want to take a break, or we can do Q&A for this? I mean, you can take a break and maybe regroup in 2 minutes. Need some time.
As you wish.
Okay, we'll just 2 minutes. We'll
okay.
You can take a catch-up break.
Okay, yeah, that works.
We'll get back to questions.
Yeah, okay.
Can you hear me at the back? Okay, yeah. We're going to take a 2-minute quick break, and then we'll get back with maybe few questions for the part that Aaron has already covered, and then we'll get on to the next part so that we stay on time. All right, so I think Aaron has done a fantastic job of holding our fingers and taking us through how the field has developed. So I'm sure there might be a few questions coming across. We'll be happy to take those. Do you have mics around you? Yeah, go ahead.
Hi, Aaron. So I was just wondering about the question of ataxia, the SCAR2 mutation in ALS. So I know in C9 mediated ALS FTD, there's accumulation, there's P62 and TDP43 positive inclusions in cerebellum, but cerebellum is not affected. I mean, the degeneration is only in the motor neurons. So is there a selectivity in only motor neurons? Do you think that is happening in case of ataxia too?
Yeah, it's a great question. So in long repeats of ataxin 2 in SCA2, there certainly is degeneration in the cerebellum. But it seems like when the intermediate repeats, the cerebellum, maybe the threshold is a little bit higher in Purkinje neurons to tolerate repeats than motor neurons, maybe. But what's weird is that the weird thing is if so, you would expect everyone with SCA2 to also have motor neuron degeneration. That's not true. We have one paper where we found a weird result where we think as you're increasing the Q length, you're stabilizing the protein. And when you're at this intermediate level, you're enhancing the function. Once you get past like the SCA2 range, now the protein's aggregating and maybe it's forming other complexes and it's doing something, some gain of function. So we think the intermediate repeats, it's more like the enhancing the normal function. The ataxia 2 ones are more like a new function due to aggregation.
Another question. Did you also screen for RNA in the TPR for C9-72? Did you also try screening only for RNA? Like it doesn't produce the protein, but only produces the RNA, and then see whether there are certain proteins which are mediating the toxicity?
For the TDP-43?
No, for C9.
Make it so it doesn't produce protein? Yes, we haven't done that, but there are some people who have done that in mouse and flies now. There's just underway. That's a good experiment. Making it so it's like interrupting it with stop codons or interrupting the translation initiation. Yeah, they're doing that now.
Thank you.
Hi, over here. Yeah, so in continuation to that, and with regard to the polyQ repeats that you found in ALS patients, I was just wondering, is this a specific type of ALS or the patients that you screened were sporadic or familiar or any specific category?
That's a great question. So I think maybe we might need to frame it as this is a new disease, a taxon 2 disease. Maybe that's why the clinical trial for 6 months didn't work. Maybe we just need to focus on individual, and maybe we need to expand the phenotype, not just, maybe it's an ataxin 2 disease. It can present with Purkinje degeneration, ataxia, or motor neuron disease. So I think where you're going with that is maybe they're not, if we look closer, they're not identical to ALS patients, other ALS patients.
We'll take one more question, if there is any.
I'd like to know why PR is more toxic than other peptides, like having arginine.
Why? Yeah, so PR and GR seem to be the most toxic, PR the most. So this thing is it's positively charged. It gets through cells, it gets into the nucleus, it gets into the nucleolus, it disrupts everything it touches. So I think it's just through some sort of cationic interactions. I think that's the main reason.
Nice.
All right, so I think we can move on to the next part. We have about another 44 minutes.
Okay, this is that picture I keep showing. You guys got right away the different hypotheses about why TDP-43 can contribute to disease. For the rest of the time, I want to talk about what's going on in the nucleus once TDP-43 is depleted and sort of a loss of function. So as soon as like the protein was identified as the key pathological protein and it was identified that it was an RNA binding protein, the first thing people in the field said was, what RNA does it bind to? So this group did experiments in mouse where they did ClipSeq, where you can pull down immunoprecipitate TDP-43 from the brain of a mouse and sequence all of the RNAs associated with it. And they found all of those. At the same time, another group did the same thing in human neurons or human brain, and they found the RNAs that it binds to, and it binds to thousands of RNAs. It seems to like UG repeats. And then there was this very important paper that came out a couple years afterwards, where they found, this is a group at Johns Hopkins, Dr. Phil Wong, and he found a major function of TDP-43 is to repress so-called cryptic exons. Cryptic exons are sequences and introns that are normally not included following splicing. And TDP-43 sits down on those yellow UG repeats, and it makes sure that these cryptic exons are not included. When you lose TDP-43, these cryptic exons get inserted. And that could be a problem for a few reasons. It might destabilize the RNA, leading it to being degraded. It might cause frame shifts. It might create new peptides that have different functions. Importantly, these are non-conserved. So if you do this experiment in mouse, you get a set of cryptic exons. If you do this in human cells, you get cryptic exons. So the function's conserved, but the specific targets are all non-conserved, so you get different ones. So if you want to find the ones relevant for human disease, you need to sort of analyze human data. So this sort of shows in cartoon form what's going on. TDP-43 is in the nucleus. TDP-43 is here. It sits down, represses this cryptic exon. Following splicing, it's not there, no problem. lose TDP-43 from the nucleus, this cryptic exon gets inserted. That could be a problem for several reasons that we just discussed. So then two labs independently, Don Cleveland's laboratory and Kevin Egan's laboratory, identified a gene called STAthmin2 as a major TDP-43 cryptic splicing target. This is a highly expressed neuronal gene. It's among the most highly expressed neuronal genes. It's a TDP-43 target. But unfortunately, in its first intron, there is a cryptic exon, exon 2A. And then there's a premature stop codon and a premature polyadenylation site. And there are TDP-43 binding sites. So exon 2A is here. Normally, this gets repressed. But when TDP-43 is absent, this cryptic exon gets inserted. And because there's a premature termination codon and a premature polyadenylation signal, you made a truncated RNA. And you can detect this in every case that has TDP-43 pathology, importantly, not those that have SOD1 mutations. And you can see from their RNA-seq data, you see this exon 2A in cases in the spinal cord that you can detect with ALS. So this cryptic exon gets inserted in ALS. But just because something is inserted and seen in disease doesn't mean it causes disease. So can you think of an experiment you could do to test if this has a function or contributes to ALS? Because lots of things are going wrong, right? How do we know this is one that's contributing to ALS? Okay, yeah. So it seems that this is downstream of TDP-43, but and it also it's very hard to actually so far detect that STAthmin2 truncated protein. We could detect the RNA though. But yeah, perhaps. But that will just tell us it's there. It won't tell us if it's actually causing the disease yet. It could, but it doesn't. This just doesn't cause. In this case, it causes you have a truncated RNA with a stop code. Yeah, you could make it right. You could remove the TDP-43 binding. See if that reverses things. People are people. Yep, exactly. You could do that. This is what these guys decided to do. This is an interesting experiment. They took human iPS cells and turned them into neurons and put them into these microfluidic chambers. And then they'll extend axons out this chamber. And then if you injure them, they'll regrow these axons. And they use several antisense oligonucleotides to knock down different genes. So if they knocked down STAthmin2, they had no axons outgrowing. So this is normal, control ASO. Injure them, the axons grow. Knock down STAthmin2, there's a defect. Knock down TDP-43, there's also a defect. Okay. Knock down STAthmin2 and then add back STAthmin2, it rescues it. That's like a control. Then the surprise was knock down TDP-43 and add back only STAthmin2 and it recovers it. So this is good evidence that the loss of this phenotype, at least the axon part of it, is due to STAthmin2 loss. So you can overexpress it. So this was pretty compelling evidence that this is probably important. They've gone on now to do that experiment where they edited the cryptic splice acceptor site in mice and stuff like that. Okay. So I want to tell you so that was like the literature. I want to tell you an experiment about in my laboratory. This was from like a brilliant graduate student, Rosa Ma at Stanford. This was like right at the beginning of COVID. So all of our labs were shut down. We couldn't like do anything. Like we just could read papers and analyze data. I mean, like what else? Like, probably the same here. And Rosa, she is pretty spectacular. So she read this paper from another laboratory, presented it at our Zoom journal club. And what this this group did, they took human ALS FTD patient brain, and they used an antibody against NuN, which detects nuclei from neurons, and they sorted those out, and they used another antibody against TDP43, and they sorted those nuclei into either a bin with TDP43 in the nucleus or without TDP43 in the nucleus. So they had all these neuronal, brain-derived neuronal nuclei with or without TDP43, and they did RNA sequencing, and they found all the changes in gene expression happening in the brain in neurons losing TDP43. And they found very interesting things, but Rosa thought she didn't care about changes in gene expression. She thought she could take their data and reanalyze it for cryptic splicing and see in the brain what are cryptic splicing events that were going on, and maybe she could find some new ones beyond the Stathman-2. So she used two new she's like sort of bilingual. She's like a computer scientist and she's a molecular biologist. She could do both. And she found 66 cryptic splicing events that happened in the brain in lacking TDP43. She found Stathman-2 again. She found POLDIP3. These were known targets of TDP43. So it was like, all right, we're on the right track. And then she's found 64 novel ones. And the one that jumped out at us is called UNC13A. This one is cool because this is the #1 GWAS hit for ALS. It's not a subtle effect. It's a very strong connection to ALS. Also to FTD with ALS, it keeps coming out. If you don't include this chromosome 9, which we now know is C9orf72, UNC13A is the top one. And this is a meta-analysis. UNC13A keeps coming out. So we've known about this for like over 15 years, but no one knew how variations in UNC13A can contribute to disease. And I think Rosa kind of figured it out. So this is the RNA-seq traces from these 7 samples that she downloaded from the web, from the other group. The other lesson here is that she got a paper in Nature just by analyzing someone else's data. And so there's all this great data online. You can find interesting things. And then it's also the importance of sharing data and making everything available. So this is exon 20, exon 21 of UNC13A. There's RNA-seq reads in the exons, 9 in the introns. That's normal. But in the cases in the neurons without TDP43, she sees these RNA-seq reads in the introns. She calls these CE for cryptic exon. So this suggests that TDP43 normally binds and represses that cryptic exon. Once we're able to get back in the lab, she can do some experiments to test that hypothesis. So she uses shRNA and knocks down TDP43 and uses quantitative RT-PCR to detect the cryptic exon. She never detects it normally, but you knock down TDP43, the cryptic exon goes up, and the full-length transcript goes down. And then by Western blot, the protein's eliminated. So this is a little bit of a different effect than with the Stathman-2. I'll say a lot more about this in a second. This was in a neuroblastoma cell line. She also did this in motor neurons differentiated from human iPS cells. 3 independent lines, knock down TDP43, the cryptic exon goes up, the full-length transcript goes down, the protein's eliminated. So UNC13A is a critical neuronal gene originally discovered in C. elegans because the worms have a defect, an uncoordinated phenotype because they have defects releasing neurotransmitters. The mouse gene is called MUNC13-1. It's discovered by my colleague at Stanford, Dr. Thomas Sudhoff. It's essential for synaptic transmission. So this is just to say that this is a very critical neuronal protein, and it seems now in the nervous system, lacking TDP43, this protein's downregulated. We then asked our colleagues around the world, can we test your brain banks? And we looked at every brain bank with TDP43 pathology. We see UNC13A cryptic exons. This is in the Mayo Clinic. This is the New York Genome Center. We can detect them with TDP43 pathology. But I wanted a way of actually visualizing these cryptic exons in single neurons in the brain of a person with ALS. So Rosa developed a single molecule fluorescence in situ hybridization approach, or BaseScope, in which she can make a probe flanking the cryptic exon and then the canonical exon 20. And she stained patient brains, and you could see this is with TDP43. So this is a nucleus that has TDP43, so there's no cryptic exon. But 1, 2, 3, 4, you can detect these red dots. This is the UNC13A foci that accumulate only in the neural nuclei lacking TDP43. As a negative control, we see lots of UNC13A, normal transcript, abundant when you have normal TP43, and lower levels of it when you lose TP43, consistent with the RT-PCR. And then remember I was saying this is like a top GWAS hit for ALS. So we just said, where is the SNPs? Like with GWAS, it's very hard to know how SNPs in a gene can affect function. So I just said, where are the SNPs? And then it turned out that the lead GWAS SNP was right in the cryptic exon itself. So UNC13A is an enormous gene. The SNPs could have been anywhere, but it was like right there. So this is always an exciting day in the laboratory because this means like, maybe we know how it's working. Maybe these SNPs increase your risk for ALS because it makes the cryptic exon more likely to be included when you lose TP43. Rosa tested that hypothesis. She's very smart. She realized that some of the samples she downloaded from the web could be from individuals that were heterozygous for the risk and the reference allele. So she had looked in her RNA-seq for allele imbalance and indeed found more risk alleles in the RNA-seq reads. And then she did this systematically across the Mayo Clinic brain bank, and she sees a dose-dependent increase in cryptic exons in the brain as you go from 0 to 1 to 2 risk alleles. And this is really crazy. This is a survival curve she generated just of humans with FTD, just separated based on their UNC13A genotype. You can see that if you have 1 risk allele, you live shorter, but you even live even shorter if you have 2 risk alleles. I don't want to overinterpret this, but I think now that we know how this works, it means that we can have at least this much survival benefit, but maybe more if we block this single cryptic exon event. So I think now we've defined a new facet of TP43 pathology, UNC13A cryptic exons, together with STATHPIN2. It adds another one, but what I think is exciting is, what about all these other ones out there? And are there profiles of these in different combinations that are playing out across the brain in different regions, different subtypes? Are there genetic variations lurking in the genome in some of these other ones? The UNC13A one was really easy. It was like right there. Like, what about other ones? We're looking for those. We think of these as like the Achilles heel. Like do you guys know about the Achilles heel? Okay, sometimes, okay. So UNC13A variants don't cause ALS, but like if something else like causes TP43 loss and you have that variant, it makes it worse. And then the obvious one, therapeutic strategies to block. Now we know like where this cryptic splice acceptor site is. We've designed an antisense oligonucleotide to sort of skip that, and we're working on that. There are other cryptic targets. They're all, interestingly, many are involved in the synapse. Cai-Wei Guo, postdoctoral fellow, just like a couple days ago, has a paper on bioRxiv where she's characterizing some additional ones. And importantly, not just showing that they're there, but doing functional studies, recording the neurons to see the consequence of these and rescuing them. I then want to talk a little bit, this is an RNA metabolism, RNA meeting, about Yi Zheng, a really amazing postdoctoral fellow. He is an RNA expert when he was a PhD student and from University of Chicago. And he came to during his interview as a postdoctoral fellow. In the interview, he started working on a project, collaborating with someone in the lab and analyzing things. And he discovered something interesting. He told me, you know, TP43 can bind other places, not just these cryptic exons. It also binds in UTRs, and maybe it regulates polyadenylation, this other facet of gene regulation. And there could be alternative polyadenylation sites, and these can lead to the same coding region, but different length UTRs that can lead to different stabilities, different localizations. And then if the alternative polyadenylation site is internal, it could lead to truncations. So Rosa reanalyzed that data set on the web. What Yi did was he like reanalyzed it. And he this time looked for changes in alternative polyadenylation. And he found evidence for widespread polyadenylation changes, alternative polyadenylation changes. He found lengthening as well as shortening of the UTRs. He reanalyzed our own in-house data sets as well as others on the web. And he then used a new technique called 3'N-seq, because some RNA-seq is an APA analysis relies on existing annotations, and he is able to get base pair resolution for the to find novel alternative polyadenylation events. And sort of many other groups sort of at the same time sort of coalesced on this in different ways. So there are some really interesting ones on here. One of them is really exciting. It's called TMEM106B. There's also VPS. There's lots of interesting things. You can look on the preprint, and then the paper's coming out soon. But I think it adds to the complexity. Loss of TP43 does a lot of interesting things. Cryptic exon inclusion, now alternative polydylation changes. This TMEM106B is exciting because it's also a risk factor for FTD, and the 3' UTR gets doubled when you lose TP43. And yeah, and not only is it a risk factor for FTD, it can form amyloid fibrils in the brain of individuals. So we're trying to see how this connects. Let me just skip to here. Okay, so I want to come back and end on this one point because it's also to talk about RNA metabolism. So not all cryptic exons are created equally. So the Stathmin-2 one, that makes a truncated RNA. There's a stop codon, and then there's a poly A signal. UNC13A, on the other hand, introduces a premature termination codon, and the protein is lost. Why do you think the protein is lost with a premature termination codon? Which pathway? Good, nonsense-mediated decay, exactly. So, nonsense-mediated decay is the cellular pathway, quality control pathway, that detects these premature stop codons. And when the ribosome is scanning along, it knocks off the exon junction complex. So you don't have NMD, and you make proteins. But if there's a premature termination codon upstream of the exon junction complex, this doesn't get knocked off, and it recruits UPF1 and SMG1, and this initiates this nonsense-mediated decay. You don't want to start making truncated proteins that could have these new functions and interfere. So it's a way of a quality control mechanism. So I had this idea. I was thinking, like, if many or most of the ones that we had found, by the way, we're analyzing the nucleus. This happens in the cytoplasm, where I said many of these are degraded by NMD. Maybe standard RNA sequencing approaches might have missed these. So I was thinking maybe there could be all these other cryptic exons out there that we just kept missing because they're getting degraded before we could see them. So back to Yi and then a research specialist, Odelia, what they did was it's a very, very simple, the simplest experiment my lab's ever done, but I think the results are exciting. They just took a small molecule inhibitor of NMD, 11J. It very rapidly inhibits NMD. So we could do this experiment in like 24 hours. Some genetic approaches you could do by knocking down UPF1 and stuff, but there you might have secondary effects that are confusing. And they just took iPS-derived neurons, and they just knocked down TP43 or didn't, inhibited NMD or didn't, and then knocked down TP43 and inhibited NMD, and just repeated the RNA sequencing. And you can see it's working. So like if you look at UNC13A, here you could detect, when you knock down TP43, you detect the cryptic exon, but it's enhanced when you inhibit NMD, suggesting that it was getting degraded by NMD. Stathmin-2, there's no increase in the cryptic exon when you inhibit NMD, consistent with it not being an NMD substrate. And now they've started to find all these NMD-sensitive cryptic exons. And for example, SELF5 is a very interesting one. This is an RNA binding protein that is very similar to TP43. It binds the same sequence. That's enhanced the cryptic exon by NMD inhibition. And by mass spec of neurons, the most downregulated protein is SELF5 when you knock down TP43, even more than Stathmin-2. We found ELOB-L3, this is a cryptic exon that was reported, but we found that inhibiting NMD enhances this cryptic exon. You can detect the protein is gone. So not only are we able to strengthen known ones, we can unmask previously unidentified cryptic exons. This is the unmasked one I'm most excited about. It's called DNMT3A, DNA methyltransferase. So it's involved in de novo methylation of DNA. We never could detect it until we knocked down TDP-43 and inhibit NMD. And then if we go back to the just looking at the protein, we see the protein's lowered. So this is a classic masked cryptic exon. And we're excited about this and trying to explore the functional significance of this. But we never would have known about it before. It's involved in de novo DNA methylation. It's expressed broadly, but interestingly, the knockout mice, they have specific neuromuscular and motor neuron degeneration. And then if you knock this out in human iPS cells, human embryonic stem cells, they have impairments in motor neurons when you differentiate them. So we're trying to figure out how this fits in. And we've developed ASOs to block this cryptic exon, and we're seeing if these can rescue some phenotypes. And then what's exciting is we've gone back to patient lines, and there's a lot of controversy in the field as with whether or not you can detect these cryptic exons in patient lines. And indeed, we can detect truncated Stathmin-2 in patient lines just under normal conditions that have a C9orf72 mutation. We can never detect the UNC13A1, and people are arguing if it's relevant. But we did this experiment where we inhibited NMD, and now we can detect this cryptic exon. And if we go back, the protein's lowered. So it was always there. We just weren't able to detect it. So some new ideas. Many cryptic exon inclusion events induce nonsense-mediated decay. We've now expanded the portfolio of TDP-43 dependent cryptic splicing targets. We think new approaches like inhibiting NMD and using proteomics could be helpful for detecting these. There's a couple new ones we're interested in, SELF5 and DNMT3A. There's evidence that TDP-43 loss itself can compromise NMD, so it gets a little bit complicated. And then I think this is a good example of survivor bias. This is like when you only select things based on a predetermined criteria. Like in World War II, analysts were like looking at airplanes that came back from bombing missions, and they were putting armor where the bullets were, but that missed all the fatal shots where like those were crashing. So like maybe like we're looking at cryptic exons that we could see, but maybe the important ones were ones that we couldn't see. And then it always seems that like other people are coming up with the same thing. So like, but what's cool is like Yi's preprint is on the same time as this other group, and this other group used a genetic approach. We used a small molecule. We came up with the same conclusion, so that's cool. These are the ideas to discuss. Those are the people to thank. Okay, I'll just put that up there for discussion topics. Happy to discuss this stuff, other stuff, anything you like. Thanks.
Thank you very much. Finishing on time. We got lots of time, actually, almost 15 minutes for discussions. So yeah, can take lots of questions.
Hi. So you talked about misregulation with respect to motor neurons, but now lately the glial cells have also been associated quite well with ALS phenotype. So do you see this misregulation in those cells as well, and what could be the impact of those?
Finding cryptic exons in muscle. And we're looking actually for the ones that get translated into peptides. And one interesting thing is those cryptic exons that can make peptides, those might be like new antigens that might activate the immune system because they're sort of foreign. So we're interested in that. But for sure, I think a new approach would be to see which cryptic exons are in oligodendrocytes, astrocytes, microglia, muscle, how they interplay. No, you're right. So most of them do, just by chance, and we verify this. Some of them, like Stathmin-2, I think Stathmin-2 is an exception that proves the rule. Stathmin-2, it's in the first, it's upstream, and it has a polyadenylation signal and a stop codon, so it makes it truncated. That one's not an NMD substrate. There are a few other ones like that don't introduce a frame shift, and they make a new peptide sequence, and we're interested in those for other reasons. So most do, but there are some that don't get degraded by NMD.
Hi. Does reduction of this cryptic variant can, by different decay pathway, can reduce the TDP-43 condensates?
We haven't looked at that. That's a good point. I hadn't thought about that. I was just thinking of TDP-43's role in the nucleus to regulate this, and it leaves the nucleus and it's no longer there. But whether that then feeds back to TDP-43, I don't know. That would be cool, but we have no evidence of that.
Another one that some dipeptide or tripeptide might be encoded by these cryptic variants can associate with this TDP-43 condensate.
Again, very cool idea. We're just in the stage of cataloging what peptides are being made. Then we have to make antibodies or come up with some way of detecting them. Then I think we'll be able to answer your question. It's a great idea.
Thank you.
So I had a query about the first part of the talk. So for the loss of function phenotypes, screening using yeast, right, for TDP-43 and FUS, given that a lot of functions are related to splicing and there is very little splicing in yeast, how much can we rely on yeast to give us factors that would be modifying the loss of function phenotypes? Do you think there's a bias that's involved there in using yeast?
Yeah, I think probably it's hard to use yeast to study the loss of TDP-43 function. That being said, I think the loss of function, when it aggregates, that is tied in, I think, to its eventual loss of function. I think, if the loss of function is sort of a general disturbance in RNA metabolism, that you can elicit in yeast. We got ataxin-2 from the yeast screen, so that seems to be relevant. So there could be other things. But yeah, intuitively, I've just been using it to study sort of this gain of function in the cytoplasm type of thing.
Do people in the field understand why yeast cells die when TDP-43 or FUS is overexpressed?
So I don't even know that, and so I have a living studying that. I think it's complicated. The hand wave argument is sort of some catastrophic general disruptions to RNA processes, but it's not satisfying.
Where's the question? Oh yeah, go ahead.
So 2 questions. So first is that you talked about NMD. So there could be that those dipeptides, they could lead to also lysosomal stallings, because then the RQC thing will also be come into play, and they could also, independent of NMD, can also decrease the mRNA. So could that be possible?
Yeah, absolutely. I still think we're at the early stage of figuring out the consequence of these.
And from your talk, one thing I did not understand, how the TDP-43 is getting out of the nucleus all the time. Is those cryptic exons doing something like taking out them from the nucleus, or it's just because it's that's what I am thinking, because how it was getting out of the nucleus and getting aggregated at the first place.
Yeah, that's the major focus of the entire field now, is figuring out what's the triggering event that causes the loss of TDP-43. It is known a few things. It's known that it does shuttle normally into and out of the nucleus. So maybe if it has this aggregation propensity, it might start building up in the cytoplasm. It's known that nucleocytoplasmic transport in general sort of loses efficiency with aging, so that might accumulate. But yeah, what the triggering event is, maybe an infection or injury or something, but we don't know what that is.
No role of the TDP-43 in the cytoplasm, at least known role.
I bet there is. Yeah, I don't know. I bet it's doing something important. There's some reports that it could, in neurons, be involved in transporting RNA granules along neurites.
Any more questions? All right, if not, then, thank you very much, Aaron. Let's give him a big round of applause for this wonderful talk. Thank you so much.